All right. So we are going to give a detailed account of the role of enzymes in the digestion of carbohydrates, as was asked in second CE test of 28th of November 2025. This question can also be framed as write an essay on the digestion of carbohydrates. Now, to write an essay on digestion of carbohydrates, you walk through with this outline. You introduce, then you now discuss in each body organ, at the mouth and the stomach, then at the small intestine where you talk about both the pancreatic enzymes, the pancreatic action, and then action and the brush burner. And then you conclude the essay by talking about the end products of carbohydrate digestion. Now, what will be my introduction here? My introduction is going to be that the digestion of carbohydrates occurs in the mouth, occurs in the stomach, and mainly, you can say mainly, or you can say almost. So that means mainly, that's majorly or almost, that means sometimes all of them in the intestine, in the small intestine. So that's a very good way to introduce that is the digestion of carbohydrates begins in the mouth, then to the stomach, and mainly or almost in the small intestine. Now, in the stomach, in the mouth, let me tell you the major 4 points you are going to see. Point number 1 is that salivary gland is rich in salivary alpha amylase. Another name for this enzyme is called thiolate. Good. Now, number 2 point you are going to talk about is that salivary amylase, the action of salivary amylase in the mouth is short-lived. Why do we say it is short-lived? Because the time that we spend chewing is quite very small for this action of this particular enzyme to work. So it's very short-lived due to a fall in time of chewing. All right. Now, what makes up the third point? The third point is that this particular amylase enzyme tell us how it acts. The amylase enzyme is going to act on alpha 1,4, but never alpha 1,6. And then lastly, you should be able to add that cooked starch can also be broken down to give you maltose. So important 4 points to note as we talk about the function in the mouth. The number 1, salivary gland is rich in salivary alpha amylase, otherwise called thiolate. Number 2, the action of salivary amylase is short-lived. And the reason for that is because of the fall in the chewing time that we have. The third, salivary amylase breaks down alpha 1,4, but never alpha 1,6. And then the fourth, cooked starch can be acted upon by these enzymes to form maltose. Now, for the stomach, the first thing you are going to say is that this salivary amylase that is being enwrapped with food will follow it through till it reaches the stomach. So you are going to say that salivary amylase action is going to prolong for about 20 to 30 minutes. Now, what becomes of the second point? The second point is that it is going to be inhibited by the acidic environment in the stomach. And for the acidic environment in the stomach, we mean a fall in pH. Alright? Now, the third point that you can also see is that in the stomach, which we know, that sucrose can also be hydrolyzed. Sucrose can be hydrolyzed. And then end by talking about the optimum pH. Optimum pH for it Optimum pH for salivary amylase is within 6 to 7 and is inhibited when pH falls to below 3 or 4. So in the stomach, again, I've told you the major 4 points. Number 1, salivary amylase action will extend for about 20 to 30 minutes. And this is stopped or inhibited by the acidic environment in the stomach or a falling pH. Sucrose can be hydrolyzed. And we have said optimum pH of salivary amylase is for about 6 to 7 pH. And this inhibited or stopped once pH falls to below 3 or 4. Now, in the small intestine here, in the small intestine, what are things to talk about? In the small intestine, what happens is this. Number 1, you are going to divide the small intestine into 4. You are going to divide small intestine into 2, sorry. You divide it into what happens because of the pancreatic amylase. And then you are going to talk about what happens because of the brush border enzyme. The brush border. Now, because of the pancreatic amylase, number 1, pancreatic amylase is similar to salivary amylase. The only reason, number 1, is going to be that it is more powerful. So I say, similar to salivary amylase, but more powerful. Now, by talking about that it is more powerful, what we mean is that it is going to be active against boiled and unboiled starch. Its action is active against boiled and unboiled starch. And also, it hydrolyzes all sugars. It will hydrolyze all sugars, except cellulose. Okay, pancreatic amylase also, it works best in an alkaline medium. And then the most important is that how did we have salivary amylase into this pancreatic amylase into the picture? Pancreatic amylase was poured. In the pancreas. Remember, it's a part of the pancreatic juice. Let me show you something here. So we have our duodenum like this. Now, at this portion here, we have our pancreas. Now, working in the center of the pancreas is what we call the main pancreatic duct that will join with the common bile ducts to form this structure that opens into the ampulla or vata. So what I'm saying is that pancreatic juice is part of this and is poured into the second part of duodenum, and that is where pancreatic amylase happens to be. So pancreatic amylase, if you can tell, was part of pancreatic amylase, came into the small intestine or the duodenum when pancreatic juice was poured into the duodenum. All right. So that is what I want you to appreciate, very necessary. So that's what happens at the pancreatic amylase there. So pancreatic amylase, I've highlighted some of the major things you should note. Now, I want to now talk about the brush border. Now, in the brush border, before we end this particular essay, what happens in the brush border? Now, in the brush border, number 1, the brush border of the small intestine, is that the brush border has a particular enzyme, and that particular enzyme is called the alpha-limiting dextrinase. Alpha-limiting dextrinase happens to be the only enzyme that breaks down alpha-1,6. So it breaks down the alpha-1,6 bonds to liberate dextrin and glucose. So alpha-limiting dextrinase, what it does, it breaks down alpha-1,6 bond to form a structure, the free glucose and limit dextrin. And limit dextrin. Now, I think I should add another point here on pancreatic amylase. Please add it for your next exam. That pancreatic amylase will break polysaccharides. It breaks polysaccharides into oligosaccharides. It breaks polysaccharides into oligosaccharides. So please add that statement there. So number one again, cross-border has an enzyme called alpha-limiting dextrinase, which happens to be the only enzyme that has alpha-1 6 bond. Secondly, remember that on breaking down on breaking down these structures using alpha-limiting dextrinase, we are going to form glucose and alpha limit and glucose and limit dextrin. Now, the cross-border has three major enzymes for the breaking down of oligosaccharides. What are the three major enzymes again? Maltase, sucrase, lactase. Okay? Now let's take it from there. What does maltase do? Maltase will break down maltose to give you glucose plus glucose. This enzyme that you see here will be active. What about sucrose? Sucrose is acted upon by sucrase to form glucose plus fructose. And then I have a lactase. It will act on lactose to form glucose plus lactase. Okay. So these are them here. So you have written to this point. So endeavor to write that pathway down. Then the last one, the end products. For the end products, you can list them again. The major end products are going to be the monosaccharides, which include glucose, fructose, and lactose. Majorly. Okay, you can also have to be wish to talk about the pentoses and all of that, but I've stressed more on that of glucose, fructose, and galactose, which are monosaccharides. All right, so this is an essay on the role of enzymes in carbohydrates digestion. In case it is praying separately, that write an essay on the digestion of carbohydrate, same information, just a change in the presentation of the question. So remember these key things that we have discussed in this video for your next exam. Until then, endeavor to like this video and then leave a comment there, and then also share this video and subscribe for more videos in the coming days and weeks. Thank you.